Dr. Forbes D. Porter is a senior investigator in the intramural research program of the Eunice Kennedy Shriver National Institute of Child Health and Human Development, NICHD-NIH. He's also the director of NICHD Molecular Genomics Core and an associate scientific director for translational science. He previously served as a program head and was the clinical director for NICHD from 2010 until 2022. Dr. Porter earned his MD and PhD from Washington University in St. Louis and subsequently trained in pediatrics and clinical genetics at St. Louis Children's Hospital. Dr. Porter formed his own basic and clinical research groups at the NIH starting in 1996. His research has been primarily focused on disorders of cholesterol homeostasis, specifically Smith-Lemli-Opitz Syndrome and Niemann-Pick disease, type C. More recently, his research group has also been involved in studies of CLN3 and creatine transport deficiency. His group's goals include to combine both basic and clinical science to understand the pathological processes contributing to these disorders and to develop and test therapeutic interventions. I can think of no expert better suited to get us started with a talk on Smith-Lemli-Opitz Syndrome. Ladies and gentlemen, Dr. Porter.
So this morning I've been tasked with giving an overview of Smith-Lemli-Opitz Syndrome, or SLOS. This will be very repetitive for families that have been with us for a while, but for new families it sort of puts Smith-Lemli-Opitz Syndrome in context. First of all, the history. Smith-Lemli-Opitz Syndrome was first described in 1964 by doctors Smith, Lemli, and Opitz. At the time, it was named RSH syndrome. This was a system that Dr. Opitz used to use the surnames of patients. We're not sure why, but it didn't work very well for SLOS, even though he objected to switching to SLOS. And it came from the initials of the last names of the three individuals who they described. In 1964, so you can see his opinion here, so-called Smith-Lemli-Opitz syndrome. So this is from their 1964 paper, and as you can see here, it's hard to totally read, but in these three individuals, they really describe the major features that define what I refer to now as classical Smith-Lemli-Opitz syndrome. They also recognized, because they had familial cases, that it was a genetic disorder. The first three individuals were males. It was much easier to make the clinical diagnosis in males because of the genital anomalies were present there and started to help you focus on maybe this is a given syndrome. Okay, nomenclature, if you go back, it's all Smith-Lemli-Opitz syndrome. As I mentioned earlier, Dr. Opitz referred to it as RSH syndrome. The foundation used to be called the RSH/SLOS Foundation, but now has changed to Smith-Lemli-Opitz Foundation. The older literature will refer to type 1 SLOS, which was really the classical disease, and then it was recognized that there are more severe variants, and that was referred to as type 2, somebody can go like this in the back if I wander away, type 2 SLOS. But this is really, it's a continuum. It really ranges from a lethal disorder with multiple major congenital anomalies or birth defects to into the normal range where I think some individuals aren't even really picked up. Okay, what is the phenotype? And when I say phenotype, that means what are the features that are found? Smith-Lemli-Opitz syndrome has a classical facial features. Not every child has all of these. So remember, it's a continuum. Your child may have some of these birth defects or may have some of these features, but definitely the kids don't have all of them. And what you see in percentages is the percentage of individuals in the NIH cohort that have these features. I recently had a chance to look at a review, and I was sort of like, oh, these numbers are way off, because in the literature, the numbers don't really represent the full continuum. So we're working on trying to get these numbers out there. Microcephaly, or small head, metopic prominence, sort of a forehead that is very prominent. Ptosis, or drooping of the eyelids, is very common. Broad nasal bridge, the small upturned nose, is really classical. Cleft palate and submucosal cleft, sort of a very minor form of a cleft palate, is common. Cleft lip is not really seen. Small mouth, or micrognathia. And then in some of the more severe kids, you can see these things, a broad alveolar ridge, that's the gums being very broad, or these little cysts under the tongue. Other issues involve the central nervous system or the brain. What is most common in terms of malformations there is what we call agenesis, or partial agenesis of the corpus callosum. The corpus callosum is this structure here in the middle of the brain, and it's where the nerves come down and communicate between the two sides of the brain. So you're seeing the nerves in your right and your left side of your brain communicate to each other, and they do it through the corpus callosum. And in the individuals with Smith-Lemli-Opitz syndrome, we can often see thinning of the corpus callosum or lack of part of the corpus callosum, what we call agenesis of the corpus callosum. And that's the most common central nervous system malformation seen in our cohort. You can have more severe forms of brain malformations. For the eye, cataracts in about 15%. We are seeing a high frequency of small or anomalous optic nerves. These are developmental issues. And there's also what we call foveal hypoplasia stage 1. This is basically an ultrasound of the retina, looking at the thickness of the retina and the layering of the cells in the retina. And what we're finding is a high number of individuals with SLOS. The fovea, or the part of the eye that really focuses, has the sharpest vision, is a little thin. The limb anomalies, everybody's familiar with the cutaneous 2-3 toe syndactyly. It gets reported at very high frequency. I think that's what geneticists key in on. If you see, I used to always say, and still do, if you see a child who's having some problem, who has 2-3 toe syndactyly, you need to test for Smith-Lemli-Opitz syndrome. Postaxial polydactyly, either of the hands or the feet. This one child doesn't have it, but you can have an extra toe or an extra finger, and it's on the postaxial, or the small digit side of the limb. Short thumbs and rhizomelic shortening in the more severe cases, shortening of part of the upper part of the arm. In boys especially, there can be malformations or birth defects of the penis. These can be hypospadias, or the meatus of the penis is not at the tip, but lower on in the penis. Or in more severe cases, you can have a chromosomal boy, 46XY, that looks like a female because the genitalia did not develop. In our cohort, cardiac malformations are reasonably high frequency, but note most of these are very mild cardiac malformations. There can be kidney and lung problems, GI problems. Colonic aganglionosis, or what's called Hirschsprung disease, is a failure of the nerves to migrate. The nerves during the development need to migrate through the colon, and if they don't get all the way to the end, you have part of the colon that can't push the stool through, and that's referred to as Hirschsprung disease. Pyloric stenosis is the exit part of the stomach. There's a muscle there that controls the exit of material from the stomach to the small intestine. And in individuals, it can become, the muscle can become hypertrophic or large, and it blocks the exit of material from the stomach. Photosensitivity, many of you know that if you take your children out into the sunlight, they'll get sunburned very easily, and that's been characterized in SOS. One of the problems that many of the parents are dealing with are really poor weight gain and decreased growth. Often you'll hear your doctors describe this as failure to thrive. It's not really failure to thrive. It's just that these kids are genetically not predisposed to grow. And a few years ago, well, more than a few years ago, now a decade ago, we published these growth curves to try to say, where is your child who has SLOS? Where is their growth pattern? So what you see here are the normal curves, and then you see the curves that we generated for individuals with SLOS. So, you know, if your child is here, okay, they're doing pretty well for a child with SLOS. But I can guarantee you your pediatrician is panicking and going, what's going on? Because they're way below the normal limits. So it's important for your pediatrician to recognize that the child has SLOS and to recognize they should not expect a normal growth pattern in many of these individuals. I think we're all aware that we have a number of individuals with SLOS that pass away. It makes sense in terms of the individuals who have a lot of major birth defects and major heart defects, et cetera. But we're also aware that there are older children that all of a sudden I get a call and they say, usually afterwards, that their child has passed away, and I think we've all seen this. We're not sure what it's coming from. We actually tried to look into this by getting death records and getting death certificates to see, okay, what are the causes that the physicians are listing. That was pretty absolutely worthless. Often the death certificate just says congenital disorder. Well, I knew that to begin with. It didn't help me. But this is a survival curve. I think the take-home message from this is that as parents, you need to plan on your child living, okay? In our cohort, we have a 75% probability of survival over 35 years of age, and this might be different from some of the counseling you might receive from somebody who's not familiar with the disease or reading older literature. As I said, we don't really totally understand this. When we look at the severity score, the severity scores are obviously higher in the individuals in our cohort who have died than individuals who were still living at the time, but there doesn't seem to be any correlation with age of death and severity score. A trend, but not very impressive correlation with increased cholesterol in the kids that live longer and decreased 7-dehydrocholesterol, but these are not very good correlations at all, or strong correlations is what I should say. Okay, there is also a behavioral phenotype. The infants, as you parents know, can be irritable. They don't necessarily like to be held. They have poor suck, absolutely lack of interest in feeding. So they frequently end up with a gastrostomy tube. They can show formula intolerance, and then weight gain can be agonizing, especially in the first few years of life. I can't imagine anything that's worse for a parent than having a baby who doesn't want to be fed, doesn't want to be held, and decides to scream all the time. Older children really have autistic traits, and I think Audrey Thurm and Elaine will be talking about this later. They often have sort of obsessive compulsive traits, especially related to food. I mean, some of you guys have stories about what your kids will eat, what they won't eat, and it can be very specific. Language impairment is really hard to deal with. Self-injurious behavior, biting and head banging, as you know, can be very common. Repetitive behavior, hyperactivity. Aberrant sleep patterns can be difficult for the family. The child may not need sleep, but the parents do. And then temperament dysregulation, I guess that's a nice euphemism for they like to throw fits. At older ages than typical. Okay, what's the genetics? This is an autosomal recessive disease. This was actually a nice figure from the SLOS website. What it means is we all have 2 copies of the SLOS gene, or DHCR7 is the name of the gene for 7-dehydrocholesterol reductase. Mom has a good copy, dad has a good copy. Both mom and dad have a copy that doesn't work well, or a bad copy. So you can have 4 kinds of kids. And we can go over this in detail if somebody wants this explained. You can have a child who is not an SLS carrier. They got both good copies, mom's good copy, dad's good copy. You can have 2 children that are carriers, just like mom and dad. They have a good copy and a bad copy. And then you have your child who has Smith-Lemli-Opitz Syndrome, who happen to get both bad copies of the gene. So it's 1 out of 4, or 25%. If you have a child with SLOS, you don't get 3 freebies. Afterwards, it's like when you flip a coin, it's 25% with each pregnancy. If you flip a coin, it's 50-50, but you can flip 10 heads in a row. Doesn't happen often, but you can do it. The DHCR7 gene is on a chromosome. We have 23 pairs of chromosomes. It happens to be on chromosome 11. The P is for the short arm of chromosome 11. It has a long arm and a short arm. If you unwind a chromosome, you get down to the DNA strand. And the DNA strand has 4 letters, A, G, C, and T. And the gene is actually split up in parts. We refer to these as exons and introns. That's an odd name. It's historic. The exons are the part that actually encodes the gene, and the introns are DNA that's stuck in the middle of the piece. So you can think about the gene as being broken up in parts, and what has to happen is the body has to cell has to cut these introns out and splice it into one continuous molecule that we refer to as RNA. RNA has is composed of basically 4 letters. It's A, G, C, and T gets converted to U, but if you remember from your biology classes. But it's 4 letters, and those 4 letters are put together into sets of 3. And you can think of the set of 3 as a word, and each of those words spells out and tells the body or the cell how to make a protein. And each word defines a specific amino acid that goes into making this protein. This is a scribble version of it called DHCR7, or 7-dehydrocholesterol reductase, and it has 475 amino acids in it. So this protein gets made from the RNA. And this is just a depiction of the DHCR7 protein, a little fancier than my scribble version. This is a model. Nobody has ever determined the actual structure of the DHCR7 protein. It's proven to be fairly difficult to do. It's a membrane. It sits in a membrane in the inside of the cell called the endoplasmic reticulum membrane, and it's really buried in there with probably 9 helixes that go back and forth across the membrane, and then it has parts that stick out into the rest of the cell and in the inside of the endoplasmic reticulum. And the endoplasmic reticulum is where cholesterol is synthesized. So this protein acts with other proteins to synthesize cholesterol. Okay, so continuing with the genetics, okay, if you look up on ClinVar, which is a program that keeps track of variants, or the old name for variants is mutations, but variants is probably the better term now. There are over 300 that are categorized as either pathogenic, we're pretty sure, or we're sure that they cause SLOS, or likely pathogenic, we're very confident that they cause SLOS. There are common pathological variants. The most common one is this one that used to be referred to as IVS8-1G to C. That one rolls off the tongue a lot faster than C964-1G to C, which is the current nomenclature. The C just refers to the coding RNA position or coding DNA position. And this is the most common variant, and it causes a splicing error. Remember when I showed you the picture of the gene? It had those introns. Oops. It had the introns here and the exons here, and the body needs to cut these introns out and put the gene back together as an RNA. And what happens in this common variant, which many of the children in the room probably have, the instruction of where to start to cut is okay, but the instruction where to end the cut has this change. That's that -1 position. One of those letters is misspelled. And instead, the body says, I can't see this, so the cell looks for something similar, and it finds what we refer to as a cryptic splice site or an incorrect splice site. And so you get this RNA formed that has 134 base pairs of the intron inserted into it. And when it goes to make a protein, it comes down here, and then it starts making gibberish, and then it actually stops. So that allele, or that mutation, is what we refer to as a null mutation. It doesn't have any function at all. Other common variants include this P for protein, T93M. This means at the 93rd position in the protein, where there's supposed to be a threonine, there is a misspelling error that causes it to substitute to give a methionine. So you can see here's the 3-letter word. It's supposed to be ACG, and instead it's ATG. So the protein gets made, but instead of the threonine, the amino acid that's supposed to be there, it has a substitute methionine. That causes the protein not to function well, and the protein probably gets degraded. It will still function if there's enough of it, but it doesn't function normally. There's other types of spelling errors. Another common type is what we refer to as a nonsense allele. This is a missense. Spelled wrong, substitute amino acid. What we refer to as a nonsense allele is, instead of this tryptophan at the 151st position of the protein, it actually encodes what we call as a stop codon. So here it's supposed to be TGG to the word spelling out put a tryptophan here. Instead there's a TAG, and that tells the cell to stop making the protein. So the protein gets made to the 151st position, then it just stops. And so you get a protein that's not as long as it's supposed to be and probably doesn't function at all. Okay, when we talk about genetics, and I said, well, one copy came from dad, one copy came from mom, there's often a feeling of guilt that we passed this on to our child. What you need to realize is we all have a set of bad genes. Everyone in this room has a set. I have a set. I just don't happen to know what mine are. I just happen to know what one of yours are. So we all have it. It's not something you did, didn't do, thought about, drank, didn't drink, smoked, or whatever during pregnancy or before pregnancy. They've also been in your family for generations and generations, okay? And that's sort of illustrated here in that you can look at these common alleles. This is the IVS8-1G to C. It probably arose in the British Isles, and it has a very distinct gradient going from high to low across Northern Europe. One of the other common alleles is this W151X, that nonsense allele I talked about, and it has the opposite gradient. T93M, the common missense allele, is frequently found in the Mediterranean region, and you find it definitely in individuals who have Hispanic heritage, especially in South America, et cetera. R404C was described in France. I think Marge described it, Dr. Marge Novacek described it in Canada, and we found a case in Louisiana. So you can see it sort of traced the French colonization of the United States. Now this is an older slide. I heard a conversation going earlier. Well, what about SLOS in African Americans? It can be found in African Americans, it can be found in Asians. This was the initial really description and ascertainment and description of the disorder. Also, bad is not always bad. You go, why do we have these bad genes? I use as an example a disorder called sickle cell disease. This is another autosomal recessive disease. It is of high frequency in individuals of African descent. It turns out that if you're a carrier for sickle cell disease, you actually are resistant to malaria. And you can see here's a map that shows the distribution of malaria, and here's the map showing the distribution of the sickle cell disease gene, and they overlap. So you have your 1 out of 4 children who receive both good copies of this hemoglobin gene, and they're susceptible to malaria. You have your child who received both copies of, and actually I should have done this, they're both the same in sickle cell disease. And that child has sickle cell disease, which is can be very severe and lethal disease. And then you have the heterozygotes who are like mom and dad who are resistant to malaria, and therefore the family as a whole, it has a better survival advantage. We don't know what's going on in Smith-Lemli-Opitz syndrome, although it's been hypothesized that because 7-dehydrocholesterol is the precursor to vitamin D, that there might be decreased vitamin D deficiency in carriers. Vitamin D deficiency leads to rickets and osteomalacia. Those are bone diseases. And because of this, this may provide a survival advantage to being a carrier for SLOS. So we all have them. They've been in your families for generations, and bad is not always bad. It's bad in the case if your child has SLOS, but carriers might be more healthier. Okay, the biochemistry. That's counting down. I'm running out of time. The biochemistry is this conversion of 7-dehydrocholesterol to cholesterol. It's a change in this one double bond in the last step of cholesterol synthesis, catalyzed by the DHCR7 enzyme. So in SLOS, you have high 7-dehydrocholesterol levels, and we'll talk a little bit more about this in the next talk, and typically low cholesterol levels. But realize cholesterol levels can be low, they can be normal, and I've actually seen them being high. So you can't rely on a cholesterol test. What's going wrong in Smith-Lemli-Opitz? There's probably toxic effects, direct toxic effects of the 7-dehydrocholesterol. These 7-dehydrocholesterol gets metabolized into sterile metabolites and oxysterols that are abnormal, and they probably all cause problems during embryonic development. You also frequently will have a deficiency of cholesterol, and cholesterol is important for steroid hormone synthesis, bile acids, cell membranes, cell communication, and development. It's a very important molecule for cellular function. I divide the pathology into two parts: the development, the birth defects, and the functional, or the sterile biochemistry, and I use the analogy of wiring. The birth defects, or the development, you can think of the nerves or the wires are not connected properly or are missing, okay? We're not going to be able to reasonably fix that. But the functional, you can think about the nerves make the connections, they're there, but they're not working. And the analogy to wiring is that connection is rusty, and so the electrical impulses don't go through it the way they should. This is likely a component of SLOS that we might be able to treat, and that's what we've been working to try to do. Okay, treatment. I'll go through this fast. Dietary cholesterol supplementation has become standard of care. The idea is we correct the cholesterol deficiency. Definitely we can correct the deficiency in peripheral organs such as the liver, and I think we all think that it makes the children healthier. The brain is a big question. Cholesterol does not cross the blood-brain barrier, and so there's no evidence directly impacts behavior or cognition. We studied this in a short-term study, and we did not find any difference in one measure of behavior on or off cholesterol. And one of the ideas is you can decrease the 7-dehydrocholesterol by providing cholesterol. Simvastatin, this is a study we did over a decade ago now. The rationale was to decrease 7-dehydrocholesterol levels, increase expression of the enzyme. It does cross the blood-brain barrier. We did a placebo-controlled trial. We had a statistically significant result in reducing dehydrocholesterol level, or trend towards decreasing cerebral spinal fluid or brain levels of 7-dehydrocholesterol. And we had a statistically significant result on one of the behavioral aspects, but I would not term this to be clinically significant, okay? So we got the statistics, we could publish it, but it's not making a significant impact on your daily lives. So I don't recommend it. It did provide a proof of concept that one potential approach to treat this disorder is to increase or stabilize the DHCR7 enzyme. Antioxidant therapy has been proposed. The rationale is that it decreases the 7-dehydrocholesterol toxic effects. The 7-dehydro, this double bond here system, is very sensitive to oxidation, and so you can get various oxysterols formed, including this one, DHCEO. And the idea is that if you reduce the DHCEO and the other aberrant oxysterols, that might be of benefit. Ellen has been running a trial since 2008, but we don't have published efficacy data on it. I think it needs to be formally investigated in a placebo-controlled trial. Does it work? Does it not work? Does it truly impact behavior? Cholic acid, I think many of the people in the room are familiar with this. Dr. Elias also just ran a 12-patient open-label study with cholic acid supplementation. It was tolerated. There was an increase in cholesterol levels between baseline and 2 months after being on cholic acid. And although this wasn't significant, it was close to significant. I think this is the most important thing that came out of that, is that it might be impacting the weight gain, and this would be huge, especially in the younger kids. Effect on cognition or behavior has not been determined, and that would be the next question to ask. And just to close, this is just a historic slide. There will be many people contributed to the work in this, but it just gives you an idea. First described in 1964. The phenotypic continuum was described in the mid-'80s, and the connection between the severe, classical and severe. Biochemical defect was identified in 1993, gene cloned in 1998. We made mouse models, rat models, and started understanding some of the aspects of the pathology. Thank you. And that doesn't show me how much time I went over. Okay. I don't know, were you planning to do questions or? Okay. Okay.
Does anyone have any questions? We have a Q&A microphone that you can walk up to, or we could also bring it to you.
I think you can hear me without it.
We also have people at home who want to hear you.
What is the likelihood of neither parent of African American descent being a carrier, but we still have an SLO child?
So, first of all, we actually looked at frequency of SLOS carriers with the common variant in individuals of African descent. These were U.S. individuals, and it was actually reasonably high. The Caucasian carrier frequency is on the order of 1%.
Jamie, can you talk into the microphone?
Yeah, sorry. The Caucasian frequency was on the order of 1%. The individuals of African heritage, it was on the order of 0.7%. Realize with the history of this country, there's what we call admixture, and so African Americans do have Caucasian DNA. At the same time, we've seen the disorder in individuals with of African heritage with other variants. So your probability as a carrier, the genetics are the same. You are a carrier. The father of your child is a carrier, okay? And you have a 1 in 4 chance with every pregnancy of having an individual with SLOS.
My daughter and her husband were both tested when we found out he has SLO. They weren't carriers.
Yep, and that's, remember where I had that graph, 25% of your children, okay, will not be carriers. And so one of the advantages of identifying the gene was to be able to do genetic counseling in siblings, et cetera. Okay. And it would be your daughter was without knowing anything, and if she didn't have SLOS, was a priori before any testing at 2/3 chance of being a carrier, because we knew she didn't have SLOS, so it was only 1 out of 3 of non-carrier. Her spouse would be at population frequency, and that would depend on ethnic heritage, et cetera, okay, but low.
Thank you for that question. Another question over here?
Okay.
We've got 2 questions. I'll get to you in just a moment. Go ahead.
So what if you have a wife that does not have SOL and you are a carrier? Can you have a normal kid or?
Oh yeah, yeah. If your wife is tested and does not have a problem in the SLOS gene, okay, the probability, now I'm a geneticist, nothing ever goes to zero, but the probability becomes very, very, very small, okay, of SLOS. Remember? Yeah, but you are carriers of other things, okay? So that's just part of life, guys. That's just part of life. So, you know, often people focus on the disorder that's in their family, but SLOS is rare. There are other things that are more common, but we just don't think about them, okay?
Thank you for that question. Here's another one.
Two questions. One is the majority of the research happening here in the States, or is there international research? And then the second question is, what is the typical age of the people that are getting tested? I am only asking, my sister is 56, so I think we're on the late range of known people with it, and I'm just trying to learn more. Thank you.
So, wait, is Samar here? Have we looked at the age of diagnosis in our cohort?
I don't think we've looked at that specifically.
Yeah.
But I think it's a range.
Yeah.
And with more genetic testing happening earlier, I think kids are being diagnosed much earlier. Oh yeah.
Yeah, sorry, my question wasn't of diagnosis, just of the research of possible, like the cholesterol and all the different possibilities to improve symptoms and stuff like that. Is that in the younger range those are being tested? Is there any testing on older people?
So, in designing a therapeutic trial, you probably select especially in a rare disease, the individuals who are more classical, but it would really depend on a design of a trial. And I actually don't know of, other than Ellen's, any active trials right now. And you probably do not have an age restriction on it.
So I'll show you some data, like in a couple of hours, on our population and when we're seeing them for the first time. So I'll show, there's a slide on that, but again, it's just for our population at the NIH.
Okay.
So, yeah.
I think in the interest of time, we'll have to move on.
Okay.